My name is Handol Kim. I'm co-founder and CEO of Variational AI. Most of the drugs that have been sold, over the counter or prescription drugs are small molecule drugs. They're generally the pills that you're used to. About 90% of the drugs that have been approved are small molecule drugs. However, biologics, whether they're antibodies or other biotechnology products, I think they account for 6 of the top 10 top-selling drugs that are available today. So biologics and small molecule therapeutics are both very important and very valuable business opportunities. However, as you'll be able to understand, one does not map to the other. And so we're very grateful to be here and to speak about what we do, which is working in the area of small molecules. We look at this concept called chemical space, which if you're a computational chemist or a cheminformatician, you might be familiar with, but it's a concept of, molecules that could be drug-like or that follow a certain set of heuristics, whether it's Lipinski's rule of 5 or various heuristics that state that out of all of the possible molecules, at about 10 to the 60, 10 to the 62 are potentially drug-like. And so this is a space known as chemical space. And the fact of the matter is that a very, 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 very small percentage of chemical space has actually been interrogated. And what we mean by that is that we have actually synthesized or know the properties of these molecules, only about 1 billion on a good day out of the 10 to 60. So really what chemical space is a universe of drug-like molecules that we know almost absolutely nothing about. What we do at Variational is we use machine learning and AI to efficiently search through this space to discover new drugs with optimized properties to redefine the economics of drug development and obviously improve patient outcomes across disease areas. So you're going to hear a lot of echoing of what some of our fellow presenters are working on in terms of multi-property optimization and what we're all trying to do. And fundamentally, we're trying to solve the problem, whether it's through biologics or small molecules, that developing drugs is extremely hard and it's extremely expensive. You may or may not be familiar with this concept called Eroom's law, which is the inverse of Moore's law, which has generally shown that the cost of developing drugs has doubled on a capitalized basis since 1949 every 9 years. So every 9 years, the cost to develop a drug, including taking into account the drugs that have failed, has seen the cost of drugs exceed about 2.6 billion US dollars right now. And there are a lot of reasons for that. But primarily, you will hear people in the industry talk about the complexity of biology. Biology is complex, and people throw that out as a statement that doesn't really have a lot of meaning, but it is extremely complex. So when we talk about biology, we talk about the target. The target of a drug is usually a protein, or maybe it's an enzyme, or maybe it's an ion channel. But there are all of these impacts, and the outcome of a drug is really to create a biological outcome that you're looking for. And in addition, I've mentioned that chemical space is massive. So you have the huge complexity of biology and the massive size of chemical space. They conspire to create a very hard problem. The way we also further carve out the problem is that when you look at a molecule, and I'll constrain it mainly to small molecules, but the function and the properties of a molecule as a drug is really based on its structure, the shape, how many rings, etc. These physical structural features determine the property of the molecule when it tries to bind to its target within the protein. So this is very true that structure determines property, but unfortunately, looking through chemical space through a structural lens is often a poor guide to predicting properties, and we'll explain the reasoning behind that. But the result is that the status quo in small molecule drug discovery is that it's dependent on structure-based exploration of chemical space. It's focused on experimentation and expert intuition. That's what all of these wonderfully experienced medicinal chemists do, but also through brute force computation in virtual screening and other in silico methods that some of the other panelists have spoken about. So that is the situation. So let me spend a little time in trying to explain why at Variational we believe that structure is a suboptimal way of determining drug properties. And I'll back up here and I'll say drug properties are things such as potency. So for example, what quantity of a drug is required to create some kind of biological outcome. Selectivity, is it going to bind only to the target, or is it promiscuous and bind to off targets and then create more problems than it helps? Is it synthesizable, which means is it some freaky Frankenstein molecule that no chemist could ever make in a lab, let alone mass produce and create a mass market drug? Is it water soluble, solubility, or hydrophobicity? What is the rating of that? What molecular weight? All of these properties determine essentially whether or not a drug is going to be approved for whatever endpoint that it's being measured for. So when we look at the space of molecules, and when you're looking when you're starting a drug discovery project, there's an old saying, an old chestnut, that the best place to find a new drug is to start with an old drug. And that simply speaks to the fact that the space of molecules, that the chemical space, is just so massive. So you start with molecules that are known to have activity on a given target, and then you look for molecules nearby in chemical space. That's one way of looking at it. And on the right, what we've done is we've taken this diagram, which is rather well known, and basically it's a PCA sort of plot of various molecules. And this is a very, very small subset, subsegment of chemical space. But RO3 is rule 3, RO5 is generally maps to Lipinski's, but you see how all of these spurs go off into different types of molecules. So what we're looking for is essentially within that blue space in the middle. What we're looking for are molecules that are in a similar area of chemical space, because those map to molecules that have structures that you believe are going to yield good properties of interest. So the problem is that you have to deal with is that only a very small segment of chemical space has been explored. Like I said, maybe 1 billion molecules out of 10 to the 60, which is effectively zero. What you're then constrained in doing is it's like looking for your car keys at night under a street lamp, and you only look at where the beam of light is because that's all you can look at. You can't effectively interrogate everything in the dark. And also that structure is often a poor predictor of properties. So let's kind of look at a very sort of simplified example of that. Is if you were to zoom in on a space here, even though here it's all characterized by this aqua blue color, if you were to view molecules, the molecules within this space by property rather than structure, it might look rather smooth with respect to structure, and that's how chemists look at chemical space. But when you look at it through the lens of property, it's far from smooth. It's highly discontinuous. And imagine that each pixel in this grid or in this square is a molecule, and that molecules with like properties have the same color. So your task then is to find drugs with similar properties efficiently. But how would you do that? Because the pink pixels, let's say, for example, that's what you're looking for, they're all dispersed. It's highly discontinuous. So finding the pink pixels efficiently would be very difficult. Now imagine this is massively larger and there's a much smaller percentage of pixels that are pink. So how would you solve this problem? Well, one of the illustrations that I'd like to use to kind of explain why this is actually relevant and this is important is here's a real world example. So on the left, you see a number of compounds of small molecules that are potent with respect to malaria. So in the middle, you see here a molecule, and on the left and then on the right and above, these molecules look extremely similar. So if you were a chemist, you'd say, okay, there's a functional group here that's different. And by and large, these are structurally quite similar. They're about as close to each other as they can be. The problem is what you're looking for is the potency. So here you've got 600 nanomolar, and that is good. That is something that you want. You want under a micromolar in order to have a good potential drug candidate. But if you go left here, and with a very, very small change in the structure, you see that, in fact, the potency then goes to 11 millimolar, which is absolutely useless, right? And you go to the right, it's 9.4 above. It's greater than 5 millimolar, et cetera, et cetera. Some are completely inactive. And these are very, very close neighbors. So what we've seen in the world of chemistry is that small differences in structure often result in very large differences in properties. And these are known as activity cliffs, right? Because the space of chemistry is discontinuous, all right? Whereas on the other side, what we've seen are there can be major differences structurally among molecules that, however, are that exhibit uniform properties. So here on the right, we see 4 molecules which are structurally quite different, And these are all effective GABA A agonists, but they're structurally diverse. However, with respect to property, they're uniform. So this might prompt the question as to, if properties are what we care about when we develop a drug, why don't we search directly in the space of properties instead of interrogating through this lens of structure? Well, because it's really hard is the simple answer. And that's where the machine learning comes in. So at Variational AI, what we've done is we're applying a bunch of methods. Primary amongst them is a generative model known as a variational autoencoder, ergo our name, variational. So if you look at the status quo right now, you're looking through a jagged space. And if you're to look at the landscape of molecules, you'd see that the space of molecules is enormous, 10 to the 60, and it's chaotic. It's highly discontinuous. Again, as we said, a small structural change can induce a very large property change. And it's extremely difficult to effectively and accurately navigate the full space. It's just too large of a space. What we do is we enable property-based search. We project a structured search space where the empirical properties of the target, of the molecule change smoothly. We use state-of-the-art machine learning, and we'll talk about our results, to create a continuous representation where then the optimization is extremely easy, because what we've done is we've created a smooth space of properties where you can do a very, very, very simple optimization, and then we project back into the space of structure. A concrete example of this is on the left, you see a heat map, right? And you see the dark color in the middle, and then it goes lighter in the middle. What we've done is we've essentially, this heat map corresponds to the arranging of molecules by property, right? On the right is what you see is the actual molecules that are being represented in the heat map. And these molecules change quite dramatically between steps. So this molecule and this molecule might not look that different, but trust me when I say they're quite different. What happens here is this is a result of us being able to project rearrange molecules smoothly with respect to property, and this is what they look like structurally. If we were to change this and make these molecules smooth with respect to structure, this heat map on the left would correspondingly devolve into TV snow that I showed earlier on. And then what we've done is we've shown how we can predict using our generative model to predict, in this case, hydrophobicity. So this is a true penalized log P, which is empirically measured, and then this is our predicted log P, and that is on one specific property. What we then do is we've, and in case it wasn't clear, you rearrange chemical space and make it smooth with respect to properties, such that, back in that earlier question about how do you find the pink pixels, well, you just rearrange the space of molecules and make them smooth with respect to property. So essentially what we do is similarly to most companies, not just presenting today, but in our space as well, is our goal is to make novel drugs faster. What we do, as everyone else does, but we all face the same challenges, is we train on existing data. So when we work with a partner or a collaborator, we use empirical or experimental data, whether it's from a high throughput screen campaign, and then or other data that a partner may have, and then we also supplement it with in silico or computational data, whether it's stocking scores, molecular dynamics, et cetera, et cetera. We use this data to train our algorithm, and then the algorithm is then able to generate novel molecules that were not in the training set, but are predicted to have the target properties that we encode into the algorithm, and do this extremely quickly. So we do this by rearranging chemical space by property and then enabling a smooth gradient descent. What this achieves is the discovery of novel molecules from unexplored areas of chemical space via multi-property optimization. So essentially what we're trying to do, which is not that dissimilar from a lot of companies that we compete with, is we're accelerating hit to lead or early stage drug discovery. What we're trying to do ultimately is to turn something that takes years and turn it into months. So right here you see within the box is you have hit identification, lead discovery, and lead optimization. Our goal is to be able to compress all three of these, and I've posed them in a serial fashion, but is to try and collapse all three of these stages into one, such that we can go from hit ID all the way to an optimized lead within a very short period of time, which then will allow us to go into animal models a lot quicker, file our IND, and then get into the clinic as quickly as possible. And we believe this to be a potentially transformative method of working. So where we started, the company was founded in September of 2019. We're machine learning people. We're not drug discovery people. So we had to learn this, and we had to. The term translation is extremely important and extremely relevant, in drug discovery and biology and chemistry. But what we did was we started from the state of the art in machine learning, and so we published results in 2019, and we presented at NeurIPS in 2019. The All Smiles paper, and that was Zach Alperstein is a co-founder of ours. Jason Rolfe is a co-founder and CTO, and Artem Cherkasov is a PI or principal investigator at the University of British Columbia and the Vancouver Prostate Center. So we published this this paper and these results where we were able to essentially optimize hydrophobicity penalized by synthesizability. This is a sort of a composite measure specifically for the purposes of benchmarking. They're completely and utterly irrelevant within the field of drug discovery, but we were able to demonstrate with a very small number of molecules tested, because we don't rely on reinforcement learning or particle swarm optimization or genetic algorithms, that with a very small number of molecules tested, we're able to optimize the heck out of this metric. Of course, for no practical reason in drug discovery, but it's nice to be able to publish a good result in a paper. Then when we got onto toxicity prediction, this is in the same paper, and this is training on the NIH's Tox21. This is an empirical data set, and we were able to publish state-of-the-art results as well. And so a link to that paper is down here at the bottom as well, and we invite you to have a look. But that was then. What we've been doing since then is we've been translating, what was effectively, an algorithm that was developed to publish a paper and get some citations into something that actually works. And when I say actually works, is validated by experimental data, which is validated in wet lab. And this is what we've been doing so far. Within a year, the period of about a year, we've been able to kick off 3 programs. One program is with Admare Bioinnovations, which is a life sciences venture. It essentially is an accelerator that provides in-kind services to biopharma companies. And the target there is TDP1. So we're wrapping that project up, and we've got some really awesome biochemical screen validation, and we're calculating some additional data that shows that actually our predictions are bearing true experimentally. So we're very excited about these results. They're happening as we speak. The other program that we're working on is for SARS-CoV-2. So 3CL Pro is the target, and we did this because this was needed, and there was a lot of attention being devoted to COVID-19, and we wanted to do our bit. But also there was program money available from the Digital Technology Supercluster, and there we collaborated with the Vancouver Prostate Center, Art Cherkasov being our PI, as well as Admare Bioinnovations. And we've actually generated some de novo molecules that are predicted to be efficacious and potent and whatnot against the target. And we're pretty excited about that. So we're starting to order some molecules there. And then the third project program is confidential, but the target is CNS. And that's something that we just started kicking off. And what's important about that is this target, obviously being CNS, it's a molecule that has to penetrate the blood-brain barrier. And so there are some very specific challenges related to that we don't see in, let's say, infectious disease when we're going after, 3CL Pro on SARS-CoV-2 or TDP1, which is engaged in DNA repair. So what we're trying to demonstrate is our broad-based applicability across disease areas and on a very target agnostic basis. And so we're excited to be working on these programs. So the team is, I'm a co-founder and CEO. Jason Rolfe is a CTO, and we Ali Sabarali and Zach Alperstein, Maran Hodabande. I mean, the team and Roz, our part-time VP Finance and Ops. We're a very small team, and we all used to work at a company called D-Wave Systems, which works in quantum computing. But we started this company specifically to go after this opportunity here. And we have a great series of advisors who take us down the path and make us understand that sometimes machine learning in, outside the context of drug discovery is, well, let's just say we've learned a lot in terms of really applying what we thought was state of the art machine learning to this endeavor of drug discovery. And we got to say that when people talk about biology being difficult, we understand it. When we talk about chemistry being very difficult, we've lived it. And so we've learned a lot, and we still have a ways to go, but we're very hopeful for the future.
So how well does your approach identify stereomoses? We know that chirality can induce huge differences in a function. A sad example is thalidomide.
Yeah. So again, this is one of the perils of relying on sort of structure as an exclusive guide to it. At the end of the day, you kind of deal you play the hand that you're dealt with. So again, here what we're trying to do is trying to not use structural similarities so much as a means of discovery of these new molecules, but we're trying to understand the relationship. In one way, Eric, and there's some other people who like Harish. One way of looking at it is thinking about it sort of as an inverse QSAR sort of method, where we start sort of at the target properties, and you define them within whatever range. Like I want less than 2 hydrogen bond donors. I want an X log P of this. I want a potency of like 10 nanomolar, et cetera, et cetera. What we do is we define the range of properties in addition to hard cutoffs, and then we train on data, and then we use that in order to find novel molecules that were not in the training set but fall within the ranges that are defined within the properties, such that we're not stuck in these local minima of structural similarities, right? So you can think of it being like scaffold hopping on a massively sort of powerful basis, where we're not constrained to steps in local search as we go through. That's one way of thinking about it. So what we hope is that we don't fall into these same sort of structural traps, where you have these structures that are very similar that end up doing bad things like thalidomide did. These are some of the concerns that are front and foremost on our minds.
Way to go! Yeah, thanks for that one there, Eric. And yeah, way to go, Handol, there with your response.
And also the model itself is robust, such that we can bias the search away from specific structures. We can make sure, for example, when it comes to things like chirality or filtering out pains, or there's a tremendous amount of stuff that can be done in order to make sure that what we output is in the space of what is desired, and the ability to search through a vast swath of chemical space really allows us to generate that novelty in a very quick period of time.
What could be the impact of these types of models in the overall drug discovery pipeline? Is it possible to integrate data from further steps like toxicity or manufacturing costs? Where could be now the bottlenecks?
Yeah, so that's a great question. That's fundamental to what we do. I mean, we use SAS, right? Synthetic accessibility scores, ERDL, or we can use synthesizability data, or we can use drug cost data. As long as there is data that we can use in order to define what it is that we're looking for. The other thing is in one of the projects, a proof of concept, we constrained the output of the molecules that our algorithm found to ones that were immediately orderable on enamine, right? Now you're constraining the output, but you're ensuring that you can find synthesizable molecules that are going to be manufacturable. In terms of toxicity, this is a big issue in the sense that if you know of a large enough, data set on toxicity that we could use to integrate within what we do, I'll buy you a steak dinner, or I'll buy you, a Beyond Meat if you don't eat meat. But the fact of the matter is we're constrained on toxicity based on the publicly available data sets. However, in collaboration with other partners, we find that these issues and challenges, potentially go away. But our goal is, the other thing is there are certain proxies on certain properties, have proxies that, like, for example, we can optimize on an arbitrary number of properties. We do 30 properties. You want to do like something as arcane as, I don't know, selectivity, to a specific binder, and well, I mean, there are a lot of things that we could do. However, what we find is that if you optimize too many properties, you're really constraining the space. So what we find is sometimes molecular weight is a good proxy for a whole swath of properties that you're looking for, or maybe hydrophobicity is another sort of proxy. So it really depends. The good thing about the machine learning method is that we're able to iterate and go through a lot of programs a lot quicker. So the
Please
question
do.
is leader in molecular machines, photochromism and electrochromism, right? Switch materials and 4D labs. So we can identify molecules that have these properties. So it's a very interesting question in the sense that when we started, we didn't look at pharma first, because I have a very healthy respect for the life sciences and biopharma. We in fact looked at materials, and we looked at electronics materials, we looked at fragrances and flavorings, we looked at polymers, we looked at synthetic biology as sort of areas that we could go in and disrupt. But the problem that we found is outside of pharma, in pharma there's data. There's not a huge amount of data, but as soon as you step out of there's zero data. It's still very much sort of bench driven. You don't see a lot of the embrace of complex computational models to generate a lot of data. I mean, what you're seeing is, and my view is a bit stale, but you look at Cubotics or Lab Hero, or a lot of the work that people like the Alan Asperu Guzik lab is doing at University of Toronto. You look at various other places, and they're doing a tremendous job of doing lab automation just so that we can actually start generating enough data so we can train these models. But outside of pharma, it's very difficult. We've spoken to quite a few of these players, and again, it's a bit stale, but we think that pharma is probably where we have the best chance of applying the generative model to doing novel molecular discovery. But it is by no means constrained to biopharma. For the drugs you discover, who owns the IP? Good question, Rish. It depends. We have several programs where we have ownership of the actual of the asset, that will result from the end of the project. Others are sort of fee for service, and we undertake these fee for service engagements in order to validate the technology. So it really depends. Right now, we're in the validation stage. We're still a small company, and although we're generating some exciting results, we'd like to be able to ensure that we can generate similar results and validated results on multiple programs, such that we can then raise a very large round and then try and become as awesome as AbCellera, and eventually try and develop our own pipeline. But right now, it depends, Harish. But eventually, we would like to own the drugs that we discover.
How many molecules do you typically screen and generate for a customer project? Do you use biophysical simulations?
So right now, we don't use biophysical simulations, but we can generate an arbitrary number of molecules, right? So we don't screen a certain number of molecules. We're not virtual screening in that sense. And it's good to see you again, Mark. We met like years ago. So and I'm so impressed and in awe of what you guys have accomplished. So it depends on the cutoffs, but in general. So for example, we generate no more than a few hundred molecules because medchems have to look at them. But these are molecules that all are all over a specific threshold on various properties that are set by the customer. So it largely depends. And in some cases, they go like, look, we score these and we give them like 60 or 70. In some cases, they want hundreds. In some cases, they're like, well, let's look at a couple of thousand and then triage from there. So it largely depends in the flow of the project. So Eric, can you use spectroscopic data to suggest or motivate you in the search in a molecular space that your algorithms didn't find? Well, so in that sense, no, we don't rely on spectroscopic data as if I understand your question correctly. But again, I'm not a drug discovery person, nor am I really a machine learning person. I'm just a business person. So